Hello and welcome to the final module in recognising and reporting tree bleeding. In this module we're going to be looking at other courses and reporting. My name's Charles Lane, I work for Ferrous Science Limited and I'm a Royal Society of Biology plant health professional and I'm one of the scientific advisors to the Observatory project. So we're now going to think about other causes of bleed-like symptoms. This is a very common symptom that you might see in the field when you're out doing your survey work, and it is one of the most frequent problems I encountered and am asked about. As you can see in this diversity of slides, there is a sort of water-soaked, slightly watery lesion on the tree. But you also notice it is frequently associated with some form of damage, whether it's an old branch or a branch tear. What you tend to see is watery, smelly, slimy, long, vertical streaks of fluid. In the summer months when it's drier, it often dries out to give a chalky white margin. What is actually happening here is anaerobic bacteria are colonising the heartwood, and as a result of that, excess fluid is being produced. This then runs down the surface of the stem to give you that very liquid, runny fluid, which then dries out to give you that chalky appearance. So this is characteristic symptoms of bacterial wetwood, which commonly is called slime flux. In this slide, we're going to look at a slightly more unusual symptom called frothing exudates, and is something that you might see on oak. Here, a wound has been colonised by bacteria or yeast, which can cause fermentation. So hence why you get a large amount of liquid, which can be very frothy and bubbly due to that fermentation activity. Again, it's a saprotrophic organism and will cause no further harm to the tree than the wound already has done. Now, as we've seen in the previous two slides, wounds are a very important part of understanding what the type of symptoms you might be seeing. So frequently this may be branch tears, where branches have just been torn off in strong winds. It could be branch stubs. Now that may actually be due to light shading, where branches have dropped off, or it could be due to poor pruning. There are also scars as well, so physical damage that may well be just to a vehicle driving past and damaging the outer bark. It may be due to vertebrate pests, something like sheep or rabbits or squirrels damaging the outer bark. And as we know, the outer bark is so important in protecting the heartwood and the sapwood of the tree. These wounds are very prone to saprotrophic colonisation by anaerobic bacteria, as we talked about before, and you may well see symptoms of wetwood and slime flux. And that is what you can see very clearly in the pictures in the middle and on the right-hand side. This picture shows a classic symptom of a very liquid, watery exudate on the surface of the tree. Now this could be confused with something like slime flux. It may even be confused with some kind of bleed. But when you look at this much more closely, you can see that the water is running out from a branch unit or actually a water pocket. It's often commonly seen after rainy weather. So if you do see a very wet solution running down a tree, look higher up in the canopy and see if there's a natural pocket where that water may have run out from. If it doesn't have a white chalky surround, again that would suggest to me it is water staining and slime flux. It certainly isn't a bleed as we've seen before by some of the other pests and pathogens. So in the next slide, we're briefly going to look about how you might record bleeding trees on the Observatory survey form. So this is just to remind you that on the Observatory survey form, you can record the presence of bleeding in 3 separate areas, whether that's on the branches, on the stem, or more commonly called the trunk, and on the stem base. You may also want to add some additional comments. So you can add any details about the symptoms. So we talked about the importance of the position, the number, but also very helpful to know whether there are any wounds or damage, as this might help us rule out common problems like slime flux or bacterial wetwood. Again, photographs are absolutely key. So again, that's why we always encourage you to take 3 types of photographs: a close-up, slightly further back, and then the entire tree in proportion. Across this module about tree bleeding, we have looked at a wide range of different symptoms. So hopefully those on the right hand side, the water-soaked lesion, the tarry spot or cankerous bleed, and the resinosus are all classic symptoms of tree bleeds. Whilst on the left hand side, hopefully now you might be able to recognise bacterial wetwood or water staining, 2 symptoms which are not really tree bleeds, but they are lookalikes. And again, with bleeding trees, it's important to remember there's a wide range of different causes. It may well be a pest, or it may well be a pathogen. And those pathogens can include fungi like Phytophthora or Armillaria, or may include bacterial pathogens as well. There's every opportunity to record the presence of bleeding trees on the observatory survey form. Again, it's really helpful to record the presence and the position on the tree. So if you remember, Armillaria, for example, really is a stem-based disease, where something like Phytophthora or great spruce bark beetle can cause bleeding much higher up the tree. So again, recording accurately the type of bleed and the position of the bleed will really help us understand what the potential cause might be. I think many of the observatory volunteers have found the Pest and Disease Forum to be a really useful way of getting advice and guidance on whether you are actually dealing with a bleed of a tree or one of these other lookalike symptoms. But if you have any doubt in your mind, and particularly if you're concerned about any of the priority pests and diseases, it's really important you report that via Tree Alert. So hopefully you've found these 3 modules to be helpful in understanding why trees might bleed. Again, I would like to thank Anna, Susie, Charlotte and David for all their help in bringing this presentation together.